Thank- Thanks, John. Well, I'm so pleased to be introducing Pandora Pound to the meeting this evening. Welcome, Pandora. Pandora is a social scientist. She has a PhD in the sociology of medicine, and she's worked in public health and in medical schools and universities for many years before becoming research director of Safer Medicines, which is a charity which is working towards moving from animal-based research to human biology-based research. Pandora is also a research fellow at the Oxford Centre for Animal Ethics. She's written many academic papers and articles and blogs on the subject of problems of using animals in medical research. And in 2023, she published Rat Trap, which is the book which is the subject of this evening's talk. And what can I say? It's a fascinating, very readable, groundbreaking book which challenges the dominant animal research paradigm on the basis that it's poor science. So the book doesn't start from the premise of the unethical treatment of animals. It starts from an evidence-based premise that it's poor science which doesn't benefit humans. So the argument is that it's in the interest of human health that medical research transitions to human models. As the subtitle of the book says, medicine has been captured by animal research and it needs to break free. So over to you, Pandora.
Thank you very much, Anne. And I should say that Anne knows the book very well because she was a magnificent proofreader for it. So you know it inside out, don't you, Anne? But thank you very much for that introduction. So, okay, I'm going to talk today about the book, Rat Trap, and also really just say a little bit about why I think animal research is not just an animal welfare issue. And so starting earlier in this year, in February, the Conservative government promised that in the summer of this year, they were going to publish a strategy to accelerate the uptake of technologies that would reduce the use of animals in science. But of course, things changed in the summer. We had a change of government, and the Labour manifesto stated that it aims to partner with scientists, industry and civil society as we work towards the phasing out of animal testing. And indeed, the government has committed to developing a roadmap to phase out animal research. And it's going to be led by Patrick Vallance, the Minister of Science, who used to be our Chief Scientific Officer throughout the pandemic. And he'll lead it alongside Minister for Animal Welfare, Sue Hayman, and others at the Home Office. But recently at the Labour Party conference, when asked about this roadmap, Sue Hayman said, "I'm trying to get a really clear picture of where the department is on all the animal welfare issues, so that I can start to pull these threads together to actually come up with something that creates a proper animal welfare strategy." Well, that got me a bit worried because the people working towards this roadmap are not working towards improving animal welfare. They're aiming to improve the science and human health by replacing animals. And so this is what I want to talk to you about tonight. It's also basically what Rat Trap is about. And so I'll just be talking to you about the fact that animal research has consequences for humans as well as animals. We'll look at the scientific limitations of animal research that's intended to have application to humans. And then look at some of the approaches we can use instead that are based on human biology. And finally look at obstacles to progress. And so we'll go in the same order really that the book goes in, and begin by exploring how we got locked into using animals in the first place, because this sort of sets the scene, and actually it's quite useful to understand how this happened. So going back to the 1800s, if you wanted to be a physiologist, someone who was interested in studying how living organisms work, there was no established career path. There were very poor career prospects, little funding. It was a very low status profession, if you could call it that. Enter Claude Bernard, who was a physiologist and also had trained in medicine. And he set out to raise the status of physiology as a profession. And he set out in his book that his aim was to use the experimental method, so basically using animal experiments, because animal experiments were seen as a very robust scientific approach, because they entailed the use of statistics, you were able to control variables, it all took place in the laboratory, so it was seen as more scientific than had been the case previously. So animal experiments were part of his strategy for raising the status of physiology as a profession. And indeed, he was actually very successful in this. And the status increased to such an extent that it was actually able to compete with disciplines, more established disciplines such as physics and chemistry. In the UK, the Physiological Society, the first meeting was in 1876, and it's still going strong. A similar thing happened with medicine. So again, Claude Bernard argued that medicine should be conducted in the laboratory, not at the patient's bedside where it had been up until then. And again, he argued in his book that studying animals under controlled laboratory conditions and then extrapolating the results to humans was much more scientific than just observing them at their bedside or in hospital or using post-mortem studies. And doctors who had already been trying to improve the status of their profession, which was also quite low, were benefiting greatly from this association with the experimental method and the association with robust science. So animal experiments became linked with medicine quite inextricably, really, and they contributed significantly to the rise of the biomedical sciences as a respectable academic discipline. But did they actually advance human medicine? I mean, certainly towards the end of his life, when he was asked, Bernard could not identify any cases where animal experimentation had actually benefited human medicine. And Anna Kingsford, who was one of the first British women to gain a medical degree, she actually asked the professor at her Faculty of Medicine why they insisted upon vivisection, and she was told, "Well, speaking for myself and my brethren of the faculty, I do not mean to say that we claim for that method of investigation that it has been of any practical utility to medical science, nor that we expect it to do so." And certainly historians agree that the rise of vivisection in the late 1800s was very closely allied to attempts to professionalise physiology and medicine, and didn't really come about through any particular medical or scientific imperative. And nevertheless, the justification for animal experiments was that it was going to benefit humans. So how did it then kind of become established over the 20th century? Well, we went from 250 experiments being recorded in the 1880s to 95,000 by 1910, and then peaking at 5.6 million in 1971. And there were many factors that led to this huge burgeoning in numbers. So there was a rise of sort of industrial-sized breeding programs to supply scientists with animals, that were healthy and very homogeneous, very standardized animals, mainly rats and mice. The two world wars created a big demand for animal studies because of chemical warfare and radiation, and there were all sorts of animal experiments into these. And then the rise of the pharmaceutical industry after the Second World War again created more demand for animals to test and develop medicines. And then there were big national institutes of medical research all over Europe and in the UK, which sprang up post-war. This was one in North London, National Institute for Medical Research. And so we get to a situation where we are today, where animal experiments are firmly embedded within our universities, our schools of medicine. Indeed, animals convert very easily into publications, which are the sort of lifeblood of universities. And so we have a situation where animal research is very much locked in to academia, industry, and even our culture, really. So the latest statistics show that we used 2.68 million animals in science in 2023. 1.47 million were for experimental procedures, and the rest were for breeding genetically modified animals. Mice are the animals most frequently used, 60%, followed by fish and rats, birds and others. And most animal research is basic research, which is conducted in universities generally. It's kind of blue skies, curiosity-driven research. Applied research, which is supposed to have direct benefit to humans, accounts for about a quarter, and then research which is conducted for regulatory reasons, so in the safety testing of medicines and devices and so on, that's 21%. So I'm going to talk about the use of animals as models for human diseases. This is when animals are used as surrogate humans in preclinical research, so that's research that's intended to have application to humans. So an animal model is basically when an animal is manipulated in various ways to make it mimic a human disease. So, for example, in stroke research, animals will have a stroke artificially induced, typically by surgery to block the middle cerebral artery. An experimental drug will then be tested on the animals, and then they see how well the animal recovers using that drug. But how well is animal research conducted? So systematic reviews are a very robust methodology for evaluating bodies of research. And up until a couple of decades ago, they hadn't been applied to animal studies. But when after 2 decades during which they had been applied, it became very clear that scientists were failing to adhere to even the most basic principles of research. So, for example, they were failing to minimize risk of bias in their studies through not using random allocation. Only about a third of scientists were randomly allocating animals to treatment or control groups. So if you don't use random allocation, basically you can put perhaps the stronger animals into the treatment group, and it will make your study outcomes look better. So this is not to say that people deliberately do this, but there's unconscious bias. And if you don't use blinded outcome assessment, then the people who are assessing the outcomes of the study may again be unconsciously biased to give the treatment animals better outcomes. So it's important to use blinding, but again, only about a third of studies used this. And these systematic reviews also revealed a failure to control for confounding variables. These are things which could confuse the findings of studies, so things like the strain of the animal, their age, what they're fed, and so on. And also failure to report the sample size calculation up front. Hardly any of the studies did this, but if you don't say how big the sample is up front, then in theory you can keep adding animals to your study until you get the answer that you're looking for. So again, good studies would deal with all these risks of bias. It was also revealed that animal studies were very poorly reported, so analyses with the best results were selected in publications. Negative studies weren't published. It was also found that these animal models didn't generalize well to humans, so very crude animal models are used to model very complex chronic human diseases. Animal samples were used that didn't represent the patient population of interest. So, for example, maybe young animals were used in studies of stroke. And again, the laboratory experiments didn't represent the clinical context. So there's a study of a treatment for stroke, which was called Tirilazad, and it was given to animals a few minutes after the stroke was induced in the animals. And it was found to be successful, but of course, if you're a human with a stroke, it can take you several hours to get to hospital. And of course, when they tried that drug in humans, it didn't work because it was too late. So all sorts of reasons why animal research is not done particularly well. And the thing is that all these biases make animal studies appear much more positive than they actually are. So you have whole bodies of animal research that appear more positive than they are. So these failures have been acknowledged by scientists. Indeed, going back to 1999, that was probably when the first attempts were made to improve the conduct of animal studies. However, 24 years later, these efforts haven't led to better human translation. There are sort of ongoing efforts to improve the conduct of animal studies, all sorts of initiatives, guidelines for reporting animal research, how it can be made more transparent, and also a kind of worrying move towards using larger animals because scientists believe that this will, believe that they're more like humans and so that they'll translate better. But unfortunately, none of these approaches actually deals with the issue of species differences. None of them take account of species differences. So I'm just going to talk for a little bit about species differences. So of course, the theory of evolution states that species may be biologically similar, and that would be due to our common ancestry. But of course, we're also likely to be significantly different biologically because we've adapted to different ecological niches. And so if you look at these finches here, they all look very similar, but in fact, their beaks are different because they've had to evolve to the particular niche that they find themselves in, and so their beaks have had to adapt to what food was available in those ecological niches. So humans and animals have much in common. We get many of the same diseases. We sometimes take the same medication. But we are all so different. We're the same and we're different. And the thing is that even very minor differences can result in very significant differences in biological processes and outcomes. And that can have real significance when it comes to developing medications, because we often do react very differently to medication. So as we said earlier, mice are the animal most commonly used in research. And again, we have many similarities, genetic, biochemical, physiological. But the lineages that led to modern rodents and primates, us, diverged around 85 million years ago. And now we've got very different life histories. You know, we're exposed to very different toxins and pathogens. We've got different microbiomes, different immune systems. We have different diets and so on. And in fact, for example, mice are between 1,000 and 100,000 times more resistant to some toxins than humans, which makes them particularly bad models for research into sepsis. So mice can help us understand processes that arose early in evolution, but they're much less useful for understanding human chronic non-communicable diseases, because these diseases have evolved through our, our lifestyles, and they're very particular. They're human diseases. So even if all the other scientific limitations were resolved, I argue that species differences is something that is insurmountable, and it's going to remain a problem. It means that animals can't reliably predict outcomes in humans. They may provide useful information about some drugs, but they won't provide useful information about others. And so this lack of reliable clinical translation has led to a failure to generate treatments for even our most common human diseases, as well as a failure to reliably safeguard us from the adverse effects of drugs, things that we're told animal research does very well. But let's have a look. So, on average, only 8% of experimental drugs, which have already shown safety and efficacy in animals, actually go on to be approved or licensed for use in humans. And the poor predictive ability of animal studies is widely acknowledged to contribute to this very low success rate. And if we just look at neurology, which has an even lower rate of success at 5.9%, this is neurology as a whole, we'll look at this in a bit more detail. So Alzheimer's disease. So you've only got to turn to the NHS website to find there's currently no cure for Alzheimer's disease, although there's medicine that can temporarily reduce the symptoms. Now this is despite decades of research using animal models of Alzheimer's disease. And it's similar with stroke. We've had stroke animal models since after the Second World War. Numerous drugs have been found successful in animals, but then when used in humans found not to be effective, and indeed some of these have actually gone on to kill humans. There's only one drug that can be used in acute stroke, and that's a clot buster called alteplase. This drug wasn't even dependent on animal models of stroke. It was actually repurposed from the field of cardiology, and only 10 to 12% of stroke patients are actually eligible for it. And furthermore, half of those who take it will remain dependent or die, so it won't have any good effect on them. So this is hardly a success story. A drug approval rate of 0.1% in stroke, and even that one drug is not a lot to write home about. And yet mortality from stroke has fallen over the last 2 decades. Well, it's clearly not due to the drugs. So what's going on here? Well, first of all, prevention. Far fewer people are smoking now. The availability of early diagnostic technologies such as MRI scanners, and the development of stroke units. So, it's not rocket science, but it works. This is basically a stroke unit is a concentration of expertise in stroke, which really does make a difference. All these animals have been used to test treatments for traumatic brain injury. They've been used in brain injury experiments where brain damage is inflicted on animals and the drug is then tested. Since the 1950s, numerous methods have been developed for inflicting brain injury on animals, most involving dropping weights on their heads, but also rapid rotation, penetrating their brains, or blowing them up. So how many approved treatments were generated for humans as a result of these experiments? Not one. So, if we go on now to look at how well animal experiments assure the safety of drugs. So in 2015, Jared Bailey and colleagues conducted a large study in which they compared animal and human toxicity data for over 2,000 drugs. So it was a big study, and it was an important study. They found that the presence of toxicity in animals does correlate, but not very, very reliably, with the presence of toxicity in humans. However, the absence of toxicity in animal tests is unable to reliably predict an absence of toxicity in humans, which, put simply, means that if a drug appears safe in animals, it can nevertheless go on to be toxic in humans. And we know this from the various tragedies that do occur. The last one probably in this country was in 2006 in North London, where 6 previously healthy young men went into multiple organ failure just minutes after receiving experimental treatment TGN 1412. This had been tested in non-human primates and a range of other animals, none of which had caused any concern. But after the tragedy, the investigation showed that the adverse reactions were the result of unpredicted biological action. So animals had completely failed to predict what would happen in humans. But even drugs that have been approved and licensed can cause adverse reactions, or at least these become evident when the drugs are used in the wider population. So troglitazone, which was a drug developed for the treatment of diabetes, was approved in the US in 1997, but just 3 years later it had to be withdrawn after reports of deaths and severe liver failure. Again, animal studies hadn't identified any cause for concern, but tests conducted subsequently on human cells and tissues showed strong indications that the drug would adversely affect the liver. If only those had been conducted instead of the animal tests, those deaths and liver failure might have been averted. So let's have a look at the sort of things that we can use instead of animals. So there are many approaches based on human biology. We've got organoids, organs on chips, artificial intelligence, mathematical modelling. We've got the omics, so exposomics, microbiomics, the genome, digital twins, and then the more familiar approaches, public health, epidemiology, prevention, clinical trials, and there are many, many more. But if we just look at a couple of these, so organoids are microscopic versions of parts of human organs. So a liver organoid would just be a microscopic version of a human liver. And you can use them to study all sorts of things, as you can see here, toxicology, drug discovery, disease modelling, and so on. Precision medicine is interesting. So, for example, samples of a tumour can be taken from a cancer patient and then be used to generate an organoid on which various chemotherapies can be tested in order to identify the one most likely to help that individual. Now, you know, that sort of personalised medicine is amazing and something that just wouldn't be possible using animals. Organs on chips are another amazing technology. So Emulate is a company that has really pioneered these, and they've got brain chips, colon chips, kidney, liver, lung chips. You can see a cross-section of one of them here. Basically, they're tiny, they're the size of a computer memory stick, and they contain sort of microscopic hollow channels, and then these are lined with living human cells. So, for example, if it was a liver chip, they'd be cells taken from a human liver. And then blood, air, and nutrients are pumped through the channels, the aim being to sort of create a home from home for the cells, to mimic the microenvironment of the human body. And a couple of years ago, a really brilliant study was done using these chips, in fact liver chips. So the authors took 27 drugs that had been judged safe for human use based on animal studies, including troglycerone, which I just mentioned. But these drugs had then gone on to cause serious adverse reactions in humans, including death and liver failure. So 27 drugs that animal studies had found safe but hadn't been safe. These drugs were then tested in 870 liver chips, and the chips were able to detect toxicity in almost 7 out of every 8 drugs that were toxic to the human liver, and this far outperformed tests in animals. And other approaches, so physiologically based pharmacokinetic modelling is a mathematical modelling technique. It can be used to predict how chemicals are absorbed, distributed, metabolized, and excreted, and therefore to replace animal studies in drug development. Another really good approach is systems biology. So this uses existing data that's now available everywhere, biological, medical, epidemiological data, and integrates it to understand biological processes as interacting systems. So that's very different from the reductionist approach that animal studies tend to rely on. And then in silico modelling, so it is already being used with great effect. So, for example, DILIsym, this software predicted that 2 migraine drugs would be toxic to the human liver, even though animal tests had raised no concerns. And this software led to their development being terminated, and presumably avoiding all sorts of adverse reactions had just animal tests been used. So these approaches, either singly but especially in combination, have the potential to utterly transform human healthcare, and they're so far-reaching in their ambition as well. But just having these approaches available isn't sufficient on their own. As well as encouraging human biology-based research, we also have to discourage and disincentivize animal studies. So we need the carrot for the human biology-based approaches and the stick as well for the animal studies. The trouble is that unraveling animal research is difficult. There are so many different aspects and they're all kind of tangled up. So the focus on the 3 R's, which tends to itself only focus on refining and improving animal studies, we need to focus on replacement, not refinement. The whole academic culture in which animal research is embedded is another obstacle. It's completely locked into our institutions at the moment. Regulation is an important obstacle because, and it's one of the key reasons why animal research continues, if we adhered to the regulations governing animal research, in fact, animal studies wouldn't be approved, but in fact, every single one is, in the UK at least. Funding needs to be redistributed. At the moment, a lot of funding is still going to animal studies and very little into human biology-based methods. There's also the efforts of the animal research lobby, which are intent on perpetuating animal research, and they're doing their best to keep animal research going. And of course, they're a vested interest. Animal research is profitable for all the sort of the industries that support it. You know, the breeders, the people who make the devices and the cages and the surgical equipment. And there's also a public perception amongst at least some part of the public that animal research is necessary for human health. So there are lots, sorry, this is my, something I drew myself, a bit wobbly. So there are lots of kind of bottom-up initiatives for tackling these problems, but what we need is a government-led roadmap to phase out animal research. We need a top-down approach. So the people kind of working in this area agree that we need kind of a dedicated minister to spearhead a roadmap. We need a properly resourced transition programme. We need to focus on science and human benefit. And we need to set realistic but ambitious goals and intermediate goals. A roadmap would need to include reform of regulations governing animal research, but also those governing the safety testing of new drugs, as these need modernising as well. And we need to divert funding away from animal models to human relevant research, so we need to disincentivise animal studies and incentivise human based research. It's just a redistribution of funding. It's not the case that we need more money. So the other thing, and what I set out to do in my book really, was to try and tackle public perceptions of animal research. So the last government commissioned survey of the public's attitude to animal research was conducted in 2018. And unfortunately, although it used to be commissioned every 2 years, it hasn't been recommissioned since then. But then at least there was still quite a large proportion of people agreeing that the use of animals for medical research purposes is important to human health, or that animal research organisations carry out work essential for human health. So as I say, this was really why I wrote Rat Trap, and I'm just going to wind up here now. So it was a book that I wrote with the general public in mind, and for that reason I tried to make it accessible and to leave out scientific jargon. I tried to make the style more popular science than academic, and I didn't include any details of animal suffering because I want people to read it. And also, it's not about animal welfare, it's about the human suffering and the poor science. And I wanted it to be affordable, so so rather than going through an academic publisher, we went through anyway, that's another story. Anyway, so this is Rat Trap. And I would really urge you to buy it, pass it on to people, get a copy for your library, send a copy to your MP, because at the moment, people who are reading it are people who already know these things. They're the people working in this field, but I really need to try and get this book out to the general public for whom I wrote it. So, yes, so thank you very much for listening. And a special thanks to Kathy Archibald, who founded the Safer Medicines Trust, and of course all the people who support the Safer Medicines Trust, because I wouldn't have been able to write this book without them.